We can, you know where I live.
No, I know.
You can come in anytime. Yeah, no problem. You can, we can, I'm easy. I totally understand that. Hospital, you can never predict what your day is going to be like. So the urinary system, what's it for? Yeah, getting rid of waste. What else besides getting rid of waste? That's not the only thing. A very, very important thing the renal system is responsible for. Regulating electrolyte and water balance. Extremely important. That is so important, there's a whole chapter on it coming. So, water balance, electrolyte balance, getting rid of wastes. As far as anatomy goes, the renal system is one of the easier systems to look at with respect to anatomy. You don't think so? Well, we'll talk about it and then you make your decision after that. There's two kidneys. The kidneys are groups of tissue where the filtering process will take place. So kidneys are what kind of organs?
It's a retro.
Mm-hmm. Retroperitoneal. What's that mean? Behind the peritoneal organs, which we just discussed in the last grouping of topics. So we have two of them. Very highly vascular. Why?
Because they need to filtrate blood.
Yeah, that's what we're filtering. We're filtering all this stuff. The mechanisms of filtration in the kidneys are filtering blood. We're filtering blood plasma. So very highly vascular because we want our blood volume to pass through this filtering system several times during the day. So we're going to see that happening in the kidney and the kidney's mechanisms. So, know the location. They are retroperitoneal organs. They are encapsulated and covered with serous membranes, very similar to all the other organs that we've discussed already. They also have little fat capsules. And what are those going to help? Where are they? Yeah, they're kind of in a really vulnerable place. So we see that there's fat there that's going to help protect them as well. Fat is not only for protection, but when we talked muscles, when we talked heart, when we talked other organ systems, fat's all also for what? Besides protection? Say it again? Yeah, energy reserves. Okay, because do we stop filtering our blood when we go to sleep? No, it's still happening. It slows down, and what we do is we concentrate our urine more, so we pull more water out of it. But that process still happens 24 hours a day, 7 days a week. So we need an energy source there as well. Gross anatomy. So, as far as it goes, we have kidneys, we have a tube that leads from the kidneys to the storage tank, and then we have a tube that leads out. There's your urinary system anatomy. So we have kidneys, and we'll talk about the details of those in a minute. What are the tubes that lead from the kidneys to the holding tank, which is the bladder? The ureters, and then the tubes that leave, or tube that leaves the bladder out of the body to dispose of the waste materials is called the urethra. And always remember that as through the body, urethra. Okay? So that's the basic anatomy of the urinary system.
Marie, can I just go back for a second? So renal
Fascia.
fascia.
What is that?
It's outer layer of
Yep, the connective tissue. Yep.
So is it serous?
Yep. Correct.
Because it's not in the pool, so that's my mind.
What is fascia, typically?
I don't know, covering.
Connective tissue.
Yeah.
Yeah.
So I just.
And then any organ system that's in a capsule that has movement associated with it has some sort of serous membrane associated with it. Okay? So, as far as where we find them, retroperitoneal, not a lot of protection. The last two ribs, which aren't doing me too much good, what are those two ribs called? Last two sets of ribs? those floating ribs. Not a lot of protection. Actually, they can really be a pain. If I hit them, they're easily broken, and they can jab into the kidneys, which is an issue. So not a lot of protection there. We have protection by those capsules. We have protection by the fluid surrounding them, and we have protection by the fat that surrounds them. So when we have a look at the kidney itself, basically what we see is a mass of tissue that has a massive blood supply that is it's responsible for filtering that. When we look at the kidney itself, we see some large arteries and veins coming into and out of. In the textbook, we see a diagram on page 958 and the path flow of blood through the renal blood vessels. I'm not going to make you memorize that, but it gives you a feel for how complex it is and how highly vascular the kidneys are. No, you don't have to know that. So, when we look at the kidney, we open it up. This is an actual kidney, and when you do your pig dissections, I want you to make sure you dissect your kidneys. You're doing it? Well, whatever. When you get to that part, make sure you dissect your kidneys because they're beautiful in the fetal pig. And you're going to see all these parts. Huh? They usually do it 2 people to a pig. But I mean, there's plenty, so if you guys want to do your own, ask him. There's plenty. So, yeah. So, when you dissect your fetal pig's kidney, it's teeny tiny, but you'll see all these parts. So it's very simple anatomy-wise. We have areas here that drain into this central region. This central region is called the Calyx region. It's in the renal pelvis. So we see calyxes draining into the renal pelvis. These areas here are called pyramids. And then this area here, kind of wrapping the outside. What's that term we've heard before? Like when we talked about the brain outside and the what's the outside regions of the cerebral hemispheres called? The cerebral cortex, right? We see that term again. The outside regions of the kidney are called the renal cortex. The middle regions of the kidney are called the renal what? Medulla. So we've heard that when we discuss the brain. The filtering units are going to lie here. And they're going to encompass part of the cortex and part of the pyramids, or medulla. And they are going to drain eventually into the renal pelvis. The renal pelvis, kidneys up here, are then going to, what's going to happen to that fluid that comes into the renal pelvis? And this is all gravity driven.
Will eliminate it.
Yeah, it's going to, that renal pelvis material is then going to come down into this part of the renal system, which is what? The ureter. Okay? And then the ureter comes into the bladder.
What's ureter? Can you please tell me what's the difference between major colics and minor colics?
You tell me. Look at the picture and tell me what the difference is between major and minor. No. Minor is smaller and comes into a larger, which is major. That's the difference. Okay? Minor is the shorter part that comes from the pyramid. Major is the larger area. So yes, see the difference?
Yeah, okay. Yeah.
And usually those terms, you know, that's synonymous with most of anatomy. The calyxes, the minors, yes.
So like, it's just pointing to that one, but all of the other ones are...
Correct.
Okay.
And all of those little darker areas are called pyramids. Okay? The business end happens in the nephrons. So the nephrons are microscopic filtering units that we're going to find encompassing the cortex and medulla regions. There's 2 different kinds of nephrons, and it has to do with how long this little area is here. This little area there is called, anybody know what this is called?
Loop.
Loop of what?
Nephron.
No.
No, it's loop
Called the loop of Henle.
Henle.
So the two different types of neurons are just going to have a different length in the loop of Henle, and that's going to help with ion and water balance. Here's how the kidneys work. It's kind of backwards, but this is the way we do it. And it is pressure driven. So osmosis and hydrostatic pressure, the water pressure in the system, is going to drive a lot of the exchange in the system. How it works is blood comes into the beginning of the nephron. The nephron starts up here. The nephron starts at the glomerular capsule. Couple of different names for that. Bowman's capsule is another name you'll hear referring to the glomerular capsule. Bowman's capsule. Or the glomerulus. All the same thing. The renal corpuscle, also another term we use to see or describe that area. But what happens is blood comes in a net of little arteries. And what we're going to do, those capillaries are fenestrated capillaries. Do you remember when we discussed capillaries?
Yeah.
What was a fenestrated capillary?
It's porous.
It's got little what?
Hole.
Yeah, got little windows, little holes. We also have tiny little cells that cover these little arteries called podocytes. And podocytes have little extensions, or have you ever heard the term pseudopod? Feet. And they move like this.
Can you write things down side of
It's
It's in a book. It's not in a book.
yes, it is in the
No,
book.
it's not side of this one.
Okay, okay.
It says food processes.
Yes, pseudopods are false feet. They are not in the book because they don't refer to the renal system. They refer to organisms that can create those extensions. So it's not part of the renal system. Amoebas. Okay? So they take their little feet and move them like this and create suction that's going to help pull out fluid as well. So between the fenestrations and the podocytes that are surrounding the arteries, we create a suction that's going to suck out most of the liquid portion of blood as it enters the beginning of the nephrons. The next part of the process is going to take that way too much fluid because we suck out way more than we should have. But this is the way the kidney works. We're going to reabsorb fluid back as it goes through a series of tubes before that fluid ends up in something called a collecting duct. What ends up in the collecting duct is going to be what ends up doing what? Yeah, we get rid of it. The collecting duct is going to flow down those little pyramid areas, and it's going to end up depositing the material that's left into the minor calyx, major calyx, renal pelvis, down the ureter, into the bladder, and eventually out the urethra. So the nephron itself is going to fine-tune the material that was removed in the beginning with the help of a whole bunch of different highly specialized cells. We're going to put back stuff that we shouldn't have taken out in the beginning part, and that beginning part of the nephron is called the proximal convoluted tubule. Why do you think we call it proximal convoluted tubule?
Proximal because it's
Close to the beginning.
capsule.
And why do we call it convoluted? Yeah, it's all squiggly. We're going to fine-tune the water balance as that fluid moves along in the loop of Henle. Descending, ascending, different things happen. But basically, the gist of it is water balance. And then we're going to take care of ion balance when we hit the last part of those squiggly tubes. That's called the distal convoluted tubule. And then whatever is left will be dumped into the collecting duct and end up as urine. So we see that there's different types of cells, different types of connections, different amounts of mitochondria as we go through the different portions of the nephron. Question?
Is reabsorption in the same time that filtration? It's happening in the same time?
In different places in the nephron, and we're going to talk about that right now. Okay? So these cells, all these different types of cells, have different functions in the total picture. Pulling everything out, putting what we took out that we shouldn't have taken out back into the circulatory system. And what's another term for that? I put back what I shouldn't have taken out. It's called reabsorption. Reabsorption back into the circulatory system. Yeah, no fancy words. So we see different cells. Now what we're going to do is talk about what happens in the different places and how those cells play a role in that function. So this is a microscopic look. Make sure when you get your microscopic slides of the kidney, you go and look for these big, very defined circles. So this is Bowman's capsule, or the glomerular capsule, or the glomerulus, which is what's inside. That region, the renal corpuscle, is the beginning of the nephron. What you see beside it histologically is those convoluted tubules. Now remember, when I make a histology slide, what do? I make a slice. So you see the nephrons, but what you're seeing is all the little tubes, cross-sections through all the little tubes, either the distal or proximal convoluted tubule all around it. So that's what you're looking at in that slide. The nephrons are of two types, and this is going to help function with water balance. So when we look at the kidney, see this line here of artery and vein that goes this way horizontally? That's basically right between the cortex and the medulla of the kidney. So when I look at it up close, I see the cortex is lighter tissue. The medulla is basically where those pyramids start. And this is where we see that artery and vein. We have two types of nephrons. One is called a cortical nephron. Why do you think we call it a cortical nephron? Most of it is where? In the cortical end. This one is called the juxtamedullary nephron, beside the medulla. Okay, and we see a huge loop of Henle here. And again, this guy is going to be more concentrated on water balance than my cortical nephron is. So we have a longer period of time to reabsorb or secrete water, depending on what has to be done for the body. Both. Depends on what I need to do. So if I have lots of this, what might I need to do? Get rid of it. If I don't have any of that, what might I need to do? Reabsorb it. So it depends on what's going on in the body. That's what the cells are going to be told to do. And who's going to tell the cells what to do? You already know this, by the way. Not necessarily the brain, indirectly the brain, but hormones. Remember when we talked about the Renin-Angiotensin Pathway, aldosterone, all those hormones you already know. Those are the guys that are going to regulate what those cells do for a living. Whether they secrete more or reabsorb more back into the circulatory system.
Yeah, but the inner part, not inner, I'm sorry. There's a two, right? It's intrinsic controls and
We're going to get there, Mila. We're getting there. We're getting there.
No, I know you're getting there, but you said only hormones. There's something else, right?
No, I didn't say only hormones. I said the nervous system indirectly too. But we're getting there. Okay? So this is the beginning part. This is the arteries that are coming in. Afferent arteriole is doing what? Coming into the where? What's this big jumbled mess? Oh, this is the glomerulus.
Red blood cells.
Okay, so red blood cells are in the capillaries. We see them here. But this is where I'm going to do what?
Pull out the water.
Yeah, this is where I'm pulling out the water. This is the beginning. This is the glomerulus. So we see that most of the fluid that we want to suck into the nephron for the process of filtration is going to start happening here. Yes, Mila?
Yeah, I'm sorry, but you're just running so fast. I cannot, pick up the information. I just want to make
Mila,
sure you understand.
can I, 1 minute? This is recorded. You can go back and listen to it again, okay?
If I have a question, I cannot ask for it.
I know, hon, but if you keep stopping me every time I try to explain something, it's going to be more confusing for you.
But it's for this topic. I'm not looking
Okay.
for something else.
Question.
Can you please explain, again, so this, what is
The glomerulus,
glomerulus? Yeah, where is start? You said
it's the beginning of the nephron,
the beginning.
beginning of the nephron, okay?
But that tube that running down, what is it?
That's the what? Which tube?
You see this square that then
No, follow it from the beginning.
Yeah.
Then we have this group of tubes. That's the what? Proximal convoluted tubule. Then we have the loop of Henle. Then we have the distal convoluted
so
tube.
this is the part of distal tube?
Yes.
Oh, okay. Yes, I didn't understand.
Okay. So this is where we're pulling out most of the materials with the help of the podocytes and the fenestrated capillaries and some of the pressure gradients that we set up here. Because of the setup of the capillaries and the nephrons, we create a differential pressure which will allow me to pull out that fluid through the windows with the help of the podocytes. So the nephron itself has 3 different functions. First, we're going to suck everything out. And where's that going to happen? In the glomerulus. Then we are going to have to put things back that we took too much out of. What's that called? Absorption. And that's going to happen if you look at the actual picture of what a nephron looks like. It's not that straight, stretched out thing. That's the one we just looked at when we looked at the cortical and juxtamedullary nephrons. So we see a tangle of tubes, and what's surrounding that tangle of tubes? The tons and tons of capillaries. So we see this process happening as fluid runs through the nephron itself. And then we fine-tune the mix. So maybe we put back some stuff or we take out a little more stuff. And that happens near the end of the nephron. In which part? What's the end of the nephron called? Distal, squiggly tubes, convoluted tubules. We might secrete a few more things. Tubular secretion. So the process of filtration involves those 3 steps. Sucking everything out, reabsorbing things, and then fine-tuning the mix. When I say fine-tune the mix, we might have to adjust the amount of hydrogen ions we get rid of to help with pH issues in the body. We might have to suck back more hydrogen ions to do the same thing. So the renal system is going to help not only with getting rid of wastes, but it also is important in water balance, and it's also very important in ion balance. And ion balance indirectly is important for pH, the concentration of hydrogen ions in solution. So these cells are going to act in different ways depending on what's going on in the body. We're going to get messages from the central nervous system, and we're going to get messages from different chemicals and hormones in different places.
Where exactly glomerular
In the glomerulus.
In the glomerulus itself.
Correct.
In the tube, but
No,
in this,
because it's glomerular filtration. So it's going to happen in the glomerulus,
So,
in Bowman's capsule, in the beginning of the nephron.
in the beginning, it's so only
In the glomerulus.
filtration happening only in this.
Correct.
parts.
So we look at this diagram, which is a nice one because it kind of shows us what we see up close in the glomerulus. And again, the capillaries that we see there are fenestrated. They have pores, which will allow fluid to pass at a rapid rate. Those podocytes, those little foot processes that we see, will help create that suction so that we can pull fluid out. So when we look at this little diagram, we see layers of filtration membrane. We're actually looking at a little cross-section of some of those capillaries. And we see that the fenestrations and the podocytes will help to pull fluid out into the glomerulus to begin the filtration process. In this little capsule, we also create pressure gradient. Now remember, when we discussed the respiratory system, why was the pressure gradient so important?
Because pressure wants to go from high pressure to low pressure.
Exactly. Things want to move. Molecules want to move from a high pressure to a low pressure. So this little capsule also sets up a pressure gradient with the help of the podocytes and the fenestrations and just the structure itself. Very similar to what we saw in the respiratory system. So this diagram is showing us, and you don't have to memorize this, but I do want you to understand that there is a pressure difference that's going to help with movement of things from the capillaries to the glomerulus because of this pressure gradient that has been set up within the glomerulus. So that's another thing that's going to help things move from the circulatory system into the glomerulus. Question?
But we need to know what is like HB,
No.
GC.
Nope.
No?
Nope. So a lot of this is review for you, believe it or not, because you know a lot about the hormones that help to regulate these cells. You've already learned them in the endocrine system. So on page 968, they talk about some of the different things associated with the filtration membrane, which we discussed. And we also have pressure gradients that are set up, which they discuss under pressure that affects filtration. Again, understand that there's a pressure gradient, a difference. And these things are all going to affect the next thing the book talks about, glomerular filtration rate. How fast things get sucked out in the glomerulus. And that's on page 966 in the textbook. So some of the things that will affect how fast things move out in the glomerulus, the beginning of filtration in the nephron, are things like net filtration pressure. So how much of a difference do we see? What might be something that could change that net filtration? How much pressure difference we see? Well, we have blood vessels there, right? Yeah, could the diameter of blood vessels affect that net filtration pressure? Yes. And we know, after our discussion of the endocrine system, that certain hormones and certain chemical messages can affect the diameter of blood vessels. So things that might affect that could affect filtration rate. The total surface area available for filtration. If I have a larger surface area, I'm going to be able to pull out more than if I have a shorter surface area. And then the permeability of the membrane itself. How fast do cells let things out or pull things back in? Again, these are all regulated by different factors, which are discussed in this diagram. So when we look at the physiological mechanisms regulating glomerular filtration rate, this diagram is going to help us narrow things down. So there are different signals that are going to affect how fast we filter materials in the nephron. There's myogenic. What's myogenic?
Muscles.
Yes, muscles. So muscle pressure, muscle release can affect that pressure gradient that we've created in the glomerulus. Tubular mechanisms, the tubes, the size of the diameter of the tubes themselves can also affect glomerular filtration rate. These two things we define as intrinsic mechanisms of glomerular filtration rate. They're things that directly regulate that filtration rate. And then we have something called extrinsic mechanisms. And this is where our friends, the hormones and the nervous system come into play. The other very important thing that the renal system is responsible for, believe it or not, with all of its regulation of ions and water, is responsible for regulating what in the system on the whole?
Blood
Pressure. Okay? So at the top of this diagram we see systemic blood pressure. So when blood pressure goes too high, the renal system is going to kick into play to help lower blood pressure. What's the quickest way to lower blood pressure?
Decrease volume.
Decrease volume in the circulatory system. And how can the renal system decrease volume in the circulatory system? Say it? Pulling more fluids out. So those mechanisms, these mechanisms in this diagram are allowing the renal system to pull more fluids out and create more urine, basically, to decrease pressure in the system. They can do the opposite when we have to do what? Increase pressure in the system. So we also have mechanisms that work both ways with respect to the renal system and its function. Okay?
So is that a dialysis when they're getting fluid out of the system? No, that's filtering it.
Yeah, and they set up the same sort of pressure gradient for blood that passes through the machine as is set up in the glomerulus of the nephron. So they're doing that same thing. They set up a pressure difference and gradient and an osmotic difference and gradient. So concentration of ions in solution and water pressure are two factors that are going to influence filtration rate. And they do that artificially with a dialysis machine. Correct. Correct. Okay, and there's so many different diseases that can affect kidney function. Diabetes, for example. When you any disease that causes something to work way harder than it should, and why do you think diabetes causes the kidneys to work way harder than it should? It's too much glucose in the system. Now, glucose is one of those things that we don't want to take out. We want to put it back. But when you have way too much glucose in your blood plasma, your kidneys are working really, really hard because they want to put all the glucose back. They can't. There's way too much. There's only a certain amount that can get back into the circulatory system before everything dumps into the collecting duct. So what might we see in a patient who has way too high a blood glucose level? We'll see what in their urine? We'll see glucose in their urine, and we'll see increased urine production because the kidneys are working twofold to try and balance that way too much glucose in the blood plasma.
Isn't it like similar to the same thing when somebody like really likes Monster, like for example Monster or Red Bull, and so they're downing these drinks left and right, and you have so many vitamins that your kidneys are working overtime. And
Correct. Yep. So everything in what? Yeah, because you tax these systems.
I already read this
When we talk about fine balancing the mix, we talk about some of these different hormones that are going to affect the nephrons to do their job for them. One of the things that we'll find is we affect the absorption rate or secretion rate of ions like sodium. Sodium is a charged particle. So water, which is a polar molecule, is attracted to these ions. So when we talk about different feedback mechanisms, we talk about some of the specifics with respect to absorption or secretion of ions like sodium, chlorine, charged particles. And that's what they're talking about here in these feedback mechanisms. We're going to affect how the cells reabsorb or secrete sodium and other charged particles. That will in turn affect water balance as well. So that's going to then affect glomerular filtration rate as well.
So we don't need to know all that mechanism in details, just in general?
No, just what I said is good. In this diagram here, we see that the cells that line the nephrons are going to be stimulated in different ways for water balance and ion balance. The nervous system, sympathetic division of the nervous system, is one of those extrinsic controls that's going to help with filtration rate in the nephron. So that's the nervous system's part. The Renin-Angiotensin-Aldosterone Mechanism is also very important in renal secretion or absorption. It's going to help stimulate different cells to do different things. You should understand the Renin-Angiotensin mechanism on page 968 in your textbook. It's in that diagram. You see, it's actually, for me, it's always easier to look at the diagram, but we see that renin and angiotensin are going to play a role in decreasing systemic blood pressure, increases aldosterone secretion. That's going to increase the reabsorption of who? Sodium. And that's going to increase what? Blood volume. That's going to then increase what? Blood pressure. So this is a nice diagram because it shows you the flow of how things work, both intrinsic and extrinsic. So understand this diagram and the mechanisms associated with it. Understand the difference between intrinsic, extrinsic, and then basically how is that going to affect water balance? What's it going to do to the cells with respect to secretion or reabsorption? Well, when in the Renin-Angiotensin pathway, what's happening? Renin is going to catalyze the formation of angiotensin II. That's going to cause an increase in the secretion of aldosterone. And of course, you already know what aldosterone does because you remember it from the endocrine system. That's going to increase reabsorption of sodium, increase blood volume, increase blood volumes, increase pressure.
So the absorption of sodium isn't drawing water to them, right? Water will flow.
Yeah, it's called obligatory water reabsorption. So when I reabsorb things like sodium into the circulatory system, because it's a charged particle and water is a polar molecule, it's going to attract it like a magnet and go back into the circulatory system. Yes? So, as far as urine formation goes, step 2 talks about tubular reabsorption. And this is where we're going to fine-tune the mix. We have to get back into the circulatory system the things that we took out that was too much. What are some of the things that were too much? Well, since we happen to be talking about diabetics, glucose was too much. We don't want to pass that in urine. We want to bring that back into the circulatory system because we need it. So that's one of the things we want to put back, and that's one of the things that's going to come back during the reabsorption process. So the reabsorption process involves pulling back through the proximal convoluted tubules, those beginning squiggle tubes right outside the glomerulus, some of the things we need to put back. So we're going to see tubular reabsorption of some ions like sodium. And sodium and its reabsorption is going to help pull some things back into the circulatory system, like water, for example. So sodium transport across that membrane is going to help with the reabsorption of water back into the circulatory system as well. We also are going to bring back some nutrients and some other ions. Again, things like amino acids, glucose. Those are things we don't want to put out in urine. Those are things we want to keep in the circulatory system because we need them to function. So that's what's going to come back during the reabsorption process. In the process of reabsorbing things like sodium, we're also going to passively reabsorb what? Water. Not on purpose. Not that the cells were directed to do that, but water is going to follow these charged particles back. What was that called again? Something absorption. Reabsorption. What was the something before the reabsorption part? Obligatory water reabsorption. Okay? So water feels obliged to follow back those charged particles. We can only reabsorb so much, and this is the problem when we have way, way too much in our blood plasma of things. So they talk a little bit about the reabsorptive capabilities of the renal tubules and the collecting duct. One of the things that's going to help us tons in this chapter, narrow down all that information, is table 25.1 on page 971. So do I want you to know this? Yes. Table 25. No, this is a diagram. It's a table 25.1 on page 971. So this tells us what's going on with the secretion reabsorption process throughout the nephron. It tells us what's happening in the proximal convoluted tubule. It tells us what's happening in the nephron, in the loop of Henle, ascending, descending, going down, coming back up, and it tells us what happens in the distal convoluted tubule. There is a little exchange happening in the beginning of the collecting ducts, and we see that in this table as well. But by the time we get to the end of the collecting ducts and into the minor calyx, we're pretty much done with that process. Where's what's left going to go? What fluid is left that makes it past the collecting duct and into the renal pelvis is going to go out as what? Waste, urine. Okay? So we fine-tune the mix, reabsorptive and secretion, and that table is going to help us understand that as well.
For the tubular reabsorption, do you want us to know the two routes, like the transcellular and paracellular?
No, don't worry about it. The next part of the textbook is going to talk about some of the different mechanisms that cause this fine mix to happen. So the distal convoluted tubules and collecting ducts on page 972 are discussed. Again, what happens there. You should know from the table what happens there. But some of the different things that affect diuresis. What's diuresis? Yeah, it's urine output is basically what that term is. Hormones, and you know these already. This is a review for you. What's antidiuretic hormone? What does it do? So what's it going to cause an increase in the nephron?
Reabsorption.
Reabsorption of what?
Sodium and water.
Sodium, and who's going to follow sodium? Water. So indirectly, antidiuretic hormone through the reabsorption of sodium is going to help with the reabsorption of water. Hold on to more water. When do I secrete antidiuretic hormone? Opposite. And when you're dehydrated, I'm sorry, I didn't hear that. Dehydrated, right? Because you don't want to lose what water you've got. So you want to hold on to, what's a diuretic? Yeah, it gets rid of excess fluid. So an antidiuretic is going to hold on to more of that fluid. Aldosterone and angiotensin, same thing. What are they going to help us do? And we saw the Renin-Angiotensin Pathway just a moment ago. What did it help to do? Back into the circulatory system. Okay, so decreased blood volume or blood pressure or high extracellular potassium concentrations are going to trigger this pathway. Certain drugs, certain metabolites, the fine-tuned part, excess little ions, controlling pH by controlling hydrogen ion concentration. That's the last part of urine formation, and that's described under step 3 of urine formation on page 972. By the way, don't forget ANP and parathyroid hormone. Those also have an effect on renal function as well. So you should know all of those on page 972 in your textbook. So we're going to fine-tune the mix by getting rid of excess or reabsorbing depending on what's going on near the end of the nephron. That table, again, is going to be a big help because it tells exactly what's going on.
Maria, do we need to know in details how exactly, let's say, sodium or chloride or something else
Nope.
passing?
Nope.
Just what is it?
Correct.
What kind of?
That on the information on that table, the whole thing. Yes?
Well, this is the details about how they pass.
Yep.
So you need to know us details.
Correct. Yep.
You just said not.
I'm sorry, I made a mistake.
Okay.
So, summary of tubular reabsorption and secretion. This is a nice one because it gives you an overview of where some of these things are passing in the process of the filtration in the nephron. So we see about 65% of the filtrate volume, everything getting pulled out in the beginning. We see some things coming back, and this is outlined in this diagram here. It's the same information that's in that table, so don't worry about repeat information. All right, here's where the scary part of the chapter comes in, and this is where, again, I'm not going to get nuts about the details of what we call the countercurrent multiplier exchanger system. But it has to do with setting up pressure gradients and concentration gradients in the filtration process that happens in the nephron. I am not going to get crazy about you knowing this in detail. So when you see page 975 and 974 and 975, I'm not going to get crazy about the details here. But this gives you an appreciation for why exchange of these things happens in the nephrons and why the juxtaglomerular nephron has more to do with water balance than the cortical nephron. So when we have to think about water balance, those guys have more of a responsibility because they have a much longer loop of Henle where that exchange, osmotic exchange, can take place. So these are all the different mechanisms involved, all the different gradients involved in exchange in the nephrons. Again, I'm not going to make you know the details of that because you'll have to know from the table where that exchange takes place. So that's covered. So the book then goes through countercurrent multiplier, countercurrent exchanger. Again, don't worry about it.
Which exactly what page?
No. That's on page 976 in your textbook. So the description of the countercurrent multiplier and the countercurrent exchanger, don't worry about it. Formation of dilute or concentrated urine is dependent on what the body needs. So if I have too many ions in my blood plasma in solution, or if I have too much water in my blood plasma or solution, that's going to dictate what type of urine I have. Believe it or not, a small urine sample can tell your doctor quite a bit about what's going on with you, because what you excrete can tell them a lot about what's floating around in your circulatory system. Excesses of things. Again, if I take a urine sample and I suspect that a patient might be diabetic, if I see glucose in their urine, big red flag. So my suspicion was correct. So formation of dilute and concentrated urine is controlled by some of the hormones that we already mentioned. For example, antidiuretic hormone when I'm dehydrated or overhydrated. When I'm overhydrated, do I want a lot of antidiuretic hormone around? No, because what do I want to do? Get rid of that excess water. So the response, your body's response to the different levels of hydration, which are monitored by your central nervous system in your blood plasma. When we talk about fluid and electrolytes in the next chapter, we're going to talk about blood's osmolality. And that's basically the concentration of dissolved substances in solution. So we're going to kind of revisit our discussion that we had way back at first semester when we talked about tonicity of solutions and what happens with respect to cells and the movement of fluid. Urea is one of the things that is a waste product. We do some recycling. Again, central nervous system medulla has osmotic receptors, osmoreceptors, that are going to be part of the communication with the renal system with respect to what it needs to do for us and all of the electrolytes that we need to get rid of. Diuretics, chemicals that are going to do what? Cause us to get rid of more fluid by doing what? Who was helping me hold on to more fluid? Antidiuretic hormone. If I have a medication that can suppress the release of what? Antidiuretic hormone, what am I going to do? I'm going to get rid of more water. So we'll see those juxtaglomerular nephrons kind of working overtime because antidiuretic hormone isn't telling them to reabsorb back water into the system. So we'll get rid of more water with medications like that. Now certain substances, let me get out of here because this is a scary diagram. Certain substances pass at a certain rate. And that has been measured through science. Things like creatinine, for example. We know how much creatinine for the average size and weight and sex of a patient that you should release in a 24-hour period. This diagram, don't worry about it. This is overhydrated. This is what antidiuretic hormone does. So we can actually do tests on patients' urine to see if they are eliminating what they should be in a 24-hour period. One of the most common ones we do is something called a creatinine clearance test. Has anybody ever had to collect a 24-hour urine before? Ooh, funsy, right? So we collect your void for 24 hours. And we do two tests. We test the amount of creatinine that was cleared in the urine, and we also test the amount of creatinine that's in the blood. There's a calculation that we can do to make sure that the kidneys are doing their job properly. to make sure that for the amount of creatinine in blood, we are clearing that proper amount in the renal system. So they talk a little bit about renal clearance and some of the different calculations associated with that. There's a chemical that we can actually administer to patients to do the same thing. It's called Inulin, and that's the one they discuss in the textbook. So we give a patient this Inulin in a certain volume. It's on page 978 under renal clearance. We give a patient a certain amount of Inulin and do the same thing. See how long it takes for the patient to clear that in their urine. Again, there's standard values that we should see as well. So that's going to test the integrity of the kidneys, and that's what a renal clearance test will do. It can also determine some different imbalances. If you look under homeostatic imbalances, they discuss renal failure. And that's when the glomerular filtration rate starts to go way, way down. So we see that some of these waste products that should be filtered out with a normal glomerular filtration rate start to build up in the patient's system. So we can do a quick blood test for things like creatinine. Another one is a substance called blood urea nitrogen, or BUN. Are these familiar for you guys in the hospitals? Okay. So these are substances we can test and get a fair assessment, fairly quick assessment, on whether or not the glomerular filtration rate is normal in our patients. When that rate starts to go way, way down, toxins build up in patient's system, and then we might eventually have to do what? Dialysis, to get out those toxins from a patient's system. No, you do not. But you do have to understand what renal clearance is and what a renal clearance test does. So, a simple urine sample can tell me a lot about a patient. So you need to understand what a normal urine sample is. A, urine is sterile. Yes? So ladies, just a little aside, for those of you who do the hover in the ladies' room, stop that. You pee on the seat. It's not nice. Urine is sterile. It is sterile. You will not get a disease from the urine, okay? Yeah. Wipe off the seat. Sit down.
But from secretions.
Secretions like?
From your reproductive organs.
Well, hopefully you won't get those on the seat. But
Yeah, but
let's not go there. I'm just saying. Urine is sterile. Yes? Okay, sit down.
Your urine, yes, but not everything.
So normal urine is sterile. Normal urine has no cells in it. Normal urine is liquid. With what in it? Lots of water, some chemical wastes, some ions, some hydrogen ions, some urea. So when I look at it, the physical characteristics of it can tell me a lot about whether I'm getting a normal urine specimen. Color and transparency, believe it or not, that's part of a urinalysis. What color is it? What color should it be?
Pale yellow.
Very pale yellow. What if it's dark, dark, dark yellow? What kind of urine? Concentrated. Multivitamins can do it too, because some of the dyes in many of those multivitamins are filtered out. Some antibiotics. Different medications can cause your urine to turn beautiful colors like blue.
Or foods,
Like beets, exactly. Or can cause your urine asparagus. Yuck. I don't like asparagus, so good. So odor, the next thing they talk about, since we're on the topic, odor also tells us a lot about urine. Urine shouldn't have a very strong smell. When it starts to smell ammonia-y, there might be some organisms present that are creating these waste products like bacteria. When you have a urinary tract infection, anybody have one of those lately? Nasty. You have a very ammonia-y smell. Don't talk about it. So it should have I love the description in the book. Fresh urine is slightly aromatic.
Yeah.
Smells like pee. pH, that's a tough one. Normally pH should be on the acidic side, but the range is huge. What we see as far as pH goes is people who have a more basic urine might also be housing some nasty little critters like a bacteria. So when you have a urinary tract infection, we do tend to see the pH hit more basic sides because some of the waste products of bacteria are on the basic side, like the ammonias. But it's a big range. Different foods can cause changes in your urine. A vegetarian, for example, eats a very alkaline diet. So we tend to see more alkaline urine. So that's a tough one. We have to look at other things besides just the pH to get a diagnosis. And then the amount of dissolved substances in solution. And the test we do for that is called specific gravity. So we're actually measuring the density of that solution. We want it to be about 1.001 to 1.035. You don't have to memorize those numbers. But the specific gravity should be more towards the waterish end.
What's the gravity?
That's an actual measurement of the density of solution. Yeah. Say it again. Correct. So when you start to see really, really high specific gravity, typically in the more concentrated urines, you'll see that as well. Exactly. Then the chemical composition. So we have wastes in there. We have urea, nitrogenous waste, creatinine. Those are all going to contribute to the pale yellow part of the urine's color. Now this here is important because this shows us what we shouldn't have in urine. These things should come back, right? They should be reabsorbed. They shouldn't make it to my urine cup. Glucose. When I start to see too much glucose in my urine, there's way too much glucose where? In my blood plasma. Diabetes might be a possible cause. Proteins. Proteins are big and typically don't get sucked out. When I start to see proteins getting sucked out, there's going to be a huge pressure gradient that's going to suck them through those fenestrations, or there might be what? If they make it into urine, there might be some kind of damage to the cells as well. So protein in our urine is also a problem. When we talk about patients with hypertension, again, huge pressure gradient there. We start to see things getting pushed out that shouldn't, or basically not being able to get back in because of the difference in pressure. You might, exactly, and usually it's associated with increase in blood pressure. Ketone bodies. Remember, too much in my blood. That means an excess of what?
Glucose.
Not
Gluconeogenesis.
Right. So when I'm breaking down too many what? Lipids at too fast a rate, that's something my liver has to deal with. If it starts to spill out into my urine, that might also be an indication of diabetes, as well as things like starvation or some of these weird diets where you're not allowed to eat any carbohydrates whatsoever. Hemoglobin in your urine. There should be no cellular material. I shouldn't say that because the urethra is lined with, anybody know? Stratified, squamous epithelial cells, and they slough off. So especially in women, sometimes your urine will be cloudy. Yes, and it's usually epithelial cells, and that's normal. Depends on which epithelial cells, though. Ketone bodies, we don't want to see them in urine. Hemoglobin, we don't want to see it in urine because hemoglobin's in urine because there was a red cell in urine at one time, and it did what? Blew up and released the hemoglobin into the urine. Biopigments, too much bilirubin, indicator of liver disease. The cells themselves, erythrocytes, not good. White blood cells, not good. That's an indication that there might be some diapedesis going on because those leukocytes want to get to your what? Urinary tract infection bacteria. So they're doing what? They're squeezing out of the nephron tubes and going into the places to try and get rid of a bacterial infection. So leukocytes might be an indicator of some sort of infection, urinary tract or bladder. So those are some of the things we don't want to see in our urine. Understand that. Urine transport, storage, and elimination. So now we go from the kidneys to the where? Ureters. Mucosa, muscularis, adventitia, very similar tubes to what we've discussed in the digestive system. Kind of like esophagus-like. We see the tube here, that's the ureter. That's going to lead to what? It's going to lead to the urinary bladder. The bladder is a holding tank. You constantly make urine. If you didn't have the bladder, what would you be doing all of the time? You would be peeing all day. So thankfully, we're designed with a holding tank. And as that holding tank fills and stretches, it gets to a certain point and then gives us a signal via the nervous system that it needs to be emptied. That process of signaling the brain is called micturition. So, urination. Urinary bladder. What kind of cells line the urinary bladder? This is a test. Stretch. It's epithelial cells.
Is it all
Yes! Good. Did you just read that? Oh, okay. Oh, I'm so excited. Transitional epithelial cells. Remember those cells that are kind of bigger at the top and smaller at the bottom in places that stretch? Exactly. So we see male and female, we see a different setup. When we discuss the reproductive system, we'll see that the exit for the renal system is shared in the male's reproductive system, and the exit, the urethra, also is uses the penis as an exit as well. In the female, we have two separate. We don't share reproductive with urinary, and our urethra is very, very short, leaving the bladder.
It's just the last part.
Correct. Yep, and that's discussed here. So when we talk about the male's reproductive system, we'll go into some of these parts discussed here as well. The prosthetic urethra, the intermediate part of the urethra, the spongy urethra, and how they relate to some of the differences in elimination with respect to the male's urinary system. Males tend not to get urinary tract infections as often as females do. How come?
Because their urethra is longer.
Much, much longer. And one of the things you do when you void is wash out the tubes. So how many ladies in this room, and I can probably pick you out, don't go to the bathroom as often as they should? Sorry. Yeah. You'll say, oh, I got to go, and then you're like, okay, I'll hold it, and then it goes away. Right? Yeah, they mess everything up. Getting old, too, does it as well. But that micturition, pay attention to that. When you get that signal to void, go.
Is this maturation?
Yep. And that's described on page 982 in the textbook. Because, huh?
I'll go all day long.
Yeah, not good.
I'll have to go like wicked bad.
Like get out of the way, I gotta go. Yeah. But see that short little tube? There's nasty things out here that can do what? Yeah, and that's exactly where they're gonna go. They're gonna crawl into your body cavity through that tube. Males is much, much longer. So make sure you void on a regular basis. That way you can decrease the incidence of urinary tract infections if you are plagued by them. That's not good, because what's happening? You know, there's some sort of degradation somewhere where the urinary system is broken down. And it depends,
So when you do menstruation,
ever had, I just went to the bathroom and I was like,
yeah.
oh my gosh.
Say it again.
So when you do menstruation or urination,
Yep.
So you kind of wash those bacteria out?
Correct. And remember, urine is what? Sterile. So, this diagram here shows us some of the controls of the response of micturition emptying the holding tank. So what are some of the things, what are some of the signals we get? That stretch reflex. Sympathetic, parasympathetic, somatic divisions of the nervous system are all involved in slowing down, speeding up, contraction of tubes.
So we need to know development aspects, right?
Nope. You need to know this, though. Okay?
Where is that?
And that is described on page 982, and the figure is on page 983. Developmental aspects, don't worry about it. We're done. Told ya.
Almost didn't make it. That clock's wrong.
When it's
Oh yeah, I got more time. I got to take attendance. So, we're done with the urinary system. We'll be on to, oops, no. Oh my crap, did I just shut that? Oh my crap. No, it's still there. Where'd it go? Okay, wait a minute. Ah. Okay, I'll figure it out. Oh, I know what I didn't shut it.